The Oxford nanopore sequencing (ONT) was developed as a technique to determine the order of nucleotides in a DNA sequence. In 2014, Oxford Nanopore Technologies released the Minion device that promises to generate longer reads that will ensure a better resolution structural genomic variants and repeat content. It's a mobile single-molecule nanopore sequencing measures 4 inches in length and is connected by a USB 3.0 port of a laptop computer. This device has been released for testing by a community of users as part of the Minion Access Program, MAP, to examine the performance of the Minion sequencer. In this sequencing technology, the first strand of a DNA molecule is linked by a hairpin to its complementary strand. The DNA fragment is passed through a protein nanopore. A nanopore is a nanoscale hole made of proteins or synthetic materials. When the DNA fragment is translated through the pore by the action of a motor protein attached to the pore, it generates a variation of an ionic current caused by differences in the moving nucleotides occupying the pore. This variation of ionic current is recorded progressively on a graphic model and then interpreted to identify the sequence. The sequencing is made on the direct strand generating the template read, and then the hairpin structure is read followed by the inverse strand generating the complement read. These reads is called 1D. If the temple and complement reads are combined, then we have a resulting consensus sequence called to direction read or to D. Among the advantages offered by this sequencer, first, its low cost and small size. Then, the sample is loaded into a port on the device and data is displayed on the screen and generated without having to wait till the run is complete. And Minion can provide very long reads exceeding 150 kbp, which can improve the contiguity of the de novo assembly. However, Minion produces a high error rate of 12%, distributed about 3% mismatches, 4% insertions, and 5% deletions. The ONT technology has continued to evolve. Recently, a new instrument has emerged called PromethION. It is the bigger brother of the Minion. It is an autonomous worktable sequencer with 48 individual flow cells, each with 3000 pores, equivalent to 48 minions, operating at 500 bp per second, which is sufficiently powerful to achieve an ultra-high throughput needed for sequencing large genomes such as the human genome. Although the PromethION is not commercially available, the ONT announces that it is capable of producing 2 to 4 TB for a duration of 2 days and a length of reads, which can attain 200 kpb, which puts this sequencer in competition with the PacBioC sequencer from Pacific Biosciences in terms of read length and HiSeq sequencer from Illumina in cost.